is Wen-Chang Lin from Academic Seneca, Taiwan. And today I will give you a brief introduction of our mobile app. We call it RNA-Seq viewer. And I think the question I often encounter when people ask why you want to develop an application to view the NGS data on a small screen iPhone or iPad, I think the simple answer is why not? Because I think today the rising demand for the mobile computing device, actually the market share of these devices outnumber the traditional desktop PC and notebooks. And so is the software development sectors. I think there are more apps showing up every day. In addition, we are using Twitter or Facebook. I think the application is tremendous. And so therefore, there are a lot of potential in the biomedical and health applications for these apps. And I think there are toolkits like health kit and research kit. And also, I think in these days, there are wearable devices like Apple Watch and Android wears. And I think these are start to shape new health management and also medical applications, especially in the clinical use and patient education sectors. So I think we started about 2 years ago. We want to know with the significance personalized medicine and advance of these sequencing big data and how we visualize and systemic display all these data on the mobile device such as iPad. And this may be helpful to have better user interface and also for the information displays. But I think the challenge is the feasibility and is there a limitation. We try to do this on this mobile device because limitation on the CPU power, and also I think the big challenge is the storage space. So we start with the TCGA data. I think this contains more than 30 some cancer types and already above 2.5 petabyte of the data. So we download about 2,500 RNA-Seq data and 4,600 microRNA-Seq data from the TCGA website. And recently we add the version 2, the RNA-Seq data. And this is the RNA-Seq viewer. It's freely available in the Apple App Stores. And the development environment is using Object-C language and also obviously with the Xcode. And the data is stored using the SQLite database. So here's a quick demo on our app. So you start with the app, and basically you see the basic illustration of human chromosome maps. So we would like to display all the data systemically on the whole chromosome view. And you can select first the sample. So you can select the specific patient sample. Here is the stomach cancer with 275 patient sample in this file. And you can change the cancer type. And then you can select the cancer of interest for studies. And then you can display the gene expression level. Tumor is in red and normal is in green. And you can see the brief clinical data on the patients, the sex, age, and the stage of the cancers. And obviously you can zoom in and zoom out and navigate around using the multi-touch. And you can find the specific region of your interest, and then you can point it. And the red hat pin will show up and point which gene you already selected. And then you can see the gene name, accession number, and the RPKM or ISMB values. And we have a sliding bar on the left, so you can just slide and you can dim the tumor expression, so you can easier to compare normal and tumor expressions. And you also can click on the NCBI button, so they will take you to the NCBI website, and you can get a better descriptions of the genes. And in this case, you can see, wow, it's quite well with the NGS data. And there's a help button. We click the I, so you take you to our website will give you a brief introductions and also the guide steps, so you can go through the introductions, how to use these apps, and also the download page. You can download the cancer of your interest, because the limitation of iPhone, iPad, we don't want to put everything into one big package. And so each cancer type is in separate files, and you can select the version 1 microRNA-Seq and also the version 2 data. But for the version 2, we only have the summarized informations. And we also provide a template, so you can download a user data tables, and you can just fill in your own experimental data on RPKM values, and then you can display using our apps. Thank you.